Well, that was an amazing introduction. Thank you all for being here in order to hear about my research experience at Lawrence, which includes exploring the inflammatory implications of common cold-induced asthma exacerbations alongside Dr. Daypal. Now, it's important to know that my research experience initially began with Dr. Sean Smith, who is also a Lawrence alum and currently working at Northwestern University, where we're basically looking at asthma epidemiology. So taking a statistical approach at trying to understand various trends in asthma exacerbations. Now, some of our interesting results are that throughout the years, 1993 up until 2011, is that the percentage mortality rate of these individuals have generally increased over the years. Interestingly enough, we also observed that race groups that account for majority of the hospitalizations belong to a lower income quartile, including African Americans and Hispanics. So what we want to take home from this is basically that further analysis is required in order to observe the associated socioeconomic factors. Improvement in the healthcare system is needed because we're seeing an increase in the percentage mortality rate, as well as that we need to develop a better understanding of asthma biophysiology because there's an increasing rate in the severity of this condition. Now, asthma exacerbations are a very prominent immune disease. On the left, you can observe a normal lung airway. However, on the right, asthma exacerbations result in the lung airway becoming inflamed due to an accumulation of mucus in the lumen, as well as due to airway remodeling. Now, symptoms associated with asthma exacerbations include coughing, chest tightness, and of course, difficulty breathing. And so some of the common triggers for asthma exacerbations include irritants such as tobacco smoke. Recent studies have shown some connections with bacterial infections, and most importantly to our research, virus infections, such as the rhinovirus. Now, rhinoviruses cause 50% of common colds. They also productively replicate in epithelial cells that are cells that line our upper respiratory tract, and they are also part of the picoRNA virus family. But what exactly are picoRNA viruses? Now, some of the most popular picoRNA viruses cause diseases such as foot and mouth disease, poliomyelitis, and hepatitis. But what this really means is that the virus will contain an outer protein coat. However, on the inside is a small RNA genome. Now, this RNA genome is single-stranded and positive sense, meaning that the whole cell ribosome can immediately hop on and result in the translation of viral protein. And just to walk you a little bit through the life cycle, rhinoviruses will bind to a receptor. They will then enter the cell, in which its RNA will become exposed. The whole cell's ribosome can immediately hop on and result in a translation of viral protein, polyprotein. However, this protein is non-functional, and so it must undergo a process known as proteolytic cleavage, meaning that it will be chopped up into functional pieces and assist with replication of the genome. From there, the genome can be packed into virions, matured, and then released from the cell. But how exactly is it that rhinoviruses are specifically connected with asthma exacerbations? Now, rhinoviruses cause what are known as a storm of inflammatory mediators. Now, this means that once rhinoviruses bind to macrophage, it will result in the release of various inflammatory mediators such as CCL18 and IL-1RN, which I will be discussing. Now, for the normal non-asthmatic individual, this means that when we have a common cold, it essentially results in us making us feel like crap. However, for someone who is asthmatic, this is more of a life or death situation because they're unable to breathe. Also, when most labs try to understand the relationship between asthma exacerbations and human rhinoviruses, they tend to focus on epithelial cells. However, in our lab, we decided to focus on the most prominent immune cell in our upper respiratory tract, which is the macrophage. Now, macrophages are basically the sentinels of the immune system. They're responsible for clearing pathogens through a process known as phagocytosis. Now, this simply means that they will surround and engulf an invading pathogen. They also present the antigens of invading pathogens, and most importantly to our research, they produce inflammatory cytokines or inflammatory mediators. Now, these inflammatory cytokines are basically, yes, responsible for bringing all the boys to the yard. They will be released from macrophages and result in the recruitment of various other immune cells to the site of infection. Now, based on that, we want to say that HRV binds to macrophages and alters the inflammatory response through the activation of various signal transduction pathways. But what are signal transduction pathways? Now, this is just basically a method in which cells are able to communicate with one another. And so it involves reception, in which a ligand, such as a hormone, will bind to its respective receptor, transduction, in which there is activation or phosphorylation of various intracellular molecules, which will finally result in the activation of some cellular response, such as cellular proliferation or apoptosis of the cell. So we can observe here that once HRV binds to its receptors, this will result in the activation of various signal transduction pathways, such as the p38 MAP kinase pathway. Now, in order for us to observe that, our experimental design included obtaining healthy human macrophages from individuals on campus. From there, we conducted various time courses where we added HRV16 or 1A at specific time intervals, and finally conducted a method which is known as a Western blot in order to observe various phosphorylation kinetics of those signal transduction pathways. Now, Western blot involves two steps. The first is an SDS page, it's just meaning that we would separate proteins in that sample based on their size. So the larger proteins are located at the top of the gel, and the smaller proteins are located at the bottom. Now, once we're able to separate those proteins based on their size, we will then transfer them onto a membrane, following which we would add a primary antibody that is specific to our protein of interest, along with a secondary antibody which is HRP linked, just meaning that it allows us to observe the relative signal intensity of that protein using software such as ImageJ. Now, what we observed in blotting for p38 MAP kinase pathway, this is some of our results that we obtained and was published in our 2014+1 paper. Now, with HRV1A, we see a gradual increase in phosphorylation kinetics up until 120 minutes. However, in HRV16, we observe that at 30 minutes, there is a peaked phosphorylation occurring and a slight decrease thereafter. GRAB2 is just simply a loading control that accounts for, yes, we loaded the same amount of sample within each well. Now, it's important to note that HRV16 and 1A are nearly genetically identical viruses. However, HRV16 is a major group rhinovirus, meaning that it binds to the ICAM1 receptor, and HRV1A is a minor group rhinovirus, meaning that it will bind to the LDL receptor. And this could possibly account for why it is that we're seeing differential phosphorylation kinetics. So once HRV1A and 16 bind to their different respective receptors, this will result in different phosphorylation kinetics being observed in the p38 MAP kinase pathway. However, this will result in the activation of the p65 or various other enzyme substrates, which can translocate into the nucleus and act as a transcription factor in order to result in various gene expressions. Now, we want to see as well if that extends with gene expression. Therefore, our collaborators at Columbia University conducted a method which is known as RNA sequencing. And RNA sequencing is basically a next generation method which allows us to sequence the entire genome of a cell. Now, what we observe here is that HRV1A and 16 compared to an untreated control is that each dot is representative of a different gene. And so the take-home message of this slide is that visually, we can observe that there's differential expression occurring between the two nearly genetically identical viruses. And so, we want to say that different viruses will result in different enzyme kinetics, as well as that viruses that bind different receptors will result in different inflammatory environments. Now, in order for us to confirm this data, we utilize a process which is known as RT-PCR, which uses fluorescence of a specific gene of interest at various cycles in order to determine the level of expression of that gene. Now, IL-1RN is the first gene that I will be discussing, and it basically encodes for an anti-inflammatory cytokine, meaning that it competitively binds to a receptor and results in suppression of the immune system, as well as ZAS3, which has been not so much studied in terms of asthma exacerbations, And I found a paper that shows that ZAS3 competitively binds with a P65, P50 dimer of the NF-kappa B signal transduction pathway. It will bind to the kappa B motif and result in a cellular apoptosis. Now, what we observe through RT-qPCR is that it's important to note at first that the minus sign is representative of a downregulation and plus sign is representative of upregulation. Now, with the addition of HRB16 and 1A, there is a decrease, a general decrease in ZAS3 expression, as well as a general decrease in IL-1RN. Now, what that is telling us is that the immune system is being activated because we're seeing a downregulation of ZAS3, which is responsible for cellular apoptosis, as well as a downregulation of IL-1RN, which is an immune suppressor. However, to me, this was the most interesting results. SB is an inhibitor of the P38 MAP kinase pathway, and we're seeing a general upregulation of ZAS3 as well as with IL-1RN. And for me, this means that inhibition of P38 MAP kinase pathway is sort of presenting itself as a potential drug target in order to assist with suppression of asthma exacerbations. So it's pretty exciting. And now, this will result, differential expression of various genes will result in possibly differential cytokine production. And so we wanted to look at CCL18 and IL-1RN cytokines. And it's important to note that, just as a reminder, they will be released from a macrophage and bind to respective receptors of various immune responsive cells. However, CCL18 is a pro-inflammatory cytokine, so it will result in the responsive cell going to the site of inflammation and assisting with fighting infection. However, IL-1RN is an immune suppressor, so it will deactivate the responsive cells. Now, in order for us to observe this, we utilize a method known as enzyme-linked immunosorbent assay. And this method basically utilizes the same method of a Western blot. However, it is more of a wet lab procedure, meaning that we're looking at protein expression within a solution. And so here we have a capture antibody that is specific for our protein of interest, along with a detection antibody and an HRP-linked antibody, meaning that we can use a substrate that will result in various color changes within solution, and from that we're able to quantify the level of or concentration of specific proteins. Now, here are results. It's important to observe that these results did not mimic RT-qPCR data. IL-1RN here, with the addition of the HRB1A and 16 rhinovirus, we see an increase in IL-1RN. However, in RT-qPCR, we saw an increase in IL-1RN. Also, here we see a decrease in IL-1RN production with the addition of the SB inhibitor. Now, basically what this is telling us is that there is possibly some mechanism that is controlling mRNA expression. And it's also very important to note that we have a very small sample size. And the same results are observed with CCL18, a very small sample size. And so this resulted in our results not being statistically significant. However, it is very interesting to observe that there are differences occurring between the two viruses. Now, in the future, we're hoping to conduct more ELISAs in order to obtain a statistically significant difference. Here is an example of an ELISA which accounts for the MCP1 protein. And the stars are annotating that there is a statistically significant difference. And there's a p-value that is less than 0.001. So that's an amazing p-value. And we're hoping to obtain results that somewhat mimic this statistical significance. So just as a brief summary, we've observed that there are different enzyme kinetics, different phosphorylation patterns. There's differential gene expression occurring within the macrophage, and as well that this results in differential cytokine production. However, there's just one more thing that I need to solve before I graduate. Now, if you can observe, there were differences between human and bovine serum cultured macrophages. So, we wanted to explore that even further with the P38, by blotting for the P38 MAP kinase protein. In human serum, we observed that there's a peak phosphorylation occurring at 30 minutes with HRB1A. However, in bovine serum, there is a gradual increase in phosphorylation occurring. So why is it that we're seeing different phosphorylation kinetics occurring between the same virus, however, in different serum? And now that got us thinking, well, bovine serum is representative of a baseline uninflamed lung. However, human serum is representative of an inflamed asthmatic lung due to a very important component known as IgG, or immunoglobulin G. Now, IgG is the most prominent antibody that is found circulating within our blood and is basically responsible for the detection of various pathogens. So therefore, we want to say that human antibodies prime macrophages in order to respond to viruses via the p38 stress kinase pathway. And so in order for us to explore that, again, we cultured macrophages in both human and bovine serum. However, we added immunoglobulin G to bovine serum cultured macrophages. From there, we conducted various time courses where we added HRV1A at specific time periods. And again, we conducted the Western blot analysis in order to observe various phosphorylation kinetics. Now here again, what we observed is that in human serum, a peak phosphorylation occurring at 30 minutes, and in bovine serum, a gradual increase, so different phosphorylation kinetics occurring. However, with addition of immunoglobulin G to bovine serum cultured macrophages, this is just one of our results that is representative of many Western blots that I've done in the past. And we generally observed that the phosphorylation kinetics of immunoglobulin G treated macrophages mimic that of human serum cultured macrophages. And so basically what that is telling us is that immunoglobulin G is very important in establishing a proper macro-inflammatory environment. From there, we wanted to add various pharmaceutical inhibitors in order to observe the same phosphorylation kinetics, including SB, which directly inhibits the p38 MAP kinase pathway, UO, which inhibits MEEK, which is responsible for the activation of ERK1 and ERK2, as well as X, which will inhibit ERK5 phosphorylation. Now, what we observed is that compared to the control, there is successful inhibition of the p38 with the addition of the SB inhibitor, as well as that there's a decrease in p38 occurring with addition of the X inhibitor of ERK5. Now, what that is telling us is that there is possibly crosstalk occurring between two pathways. However, this inhibition is not very successful in bovine serum cultured macrophages. Now, what this is basically telling us is that in future studies, researchers need to take into consideration these type of serum that they're using, as well as if they're possibly hoping to develop certain pharmaceutical inhibitors that could help in assisting with asthma exacerbations. And so we want to see, because that there's crosstalk occurring between the two pathways, we wanted to say that human antibodies prime macrophages to respond to pathogens such as viruses and bacteria via the NF-kappa B signal transduction pathway. And the NF-kappa B signal transduction pathway basically includes that phosphorylation of the I-kappa alpha inhibitor would result in the inhibitor being degraded, and the dimer, which includes P65 and P50, will translocate into the nucleus, bind to its respective motif, and result in gene expression. And if you can recall, ZAS3 competitively binds with this dimer. Now, what we observed in blotting for the P65 transcription factor is that with HRV1A, there's a gradual increase in activation. However, with HRV16, throughout the time course, we saw that there was peaked phosphorylation occurring between 15 and 30 minutes. We also observed differential phosphorylation kinetics, not only in human serum, but in bovine serum as well. There's a peaked phosphorylation occurring all throughout the time course. So again, this is telling us that immunoglobulin G is very important in establishing a proper microinflammatory environment so that we can see these varied phosphorylation kinetics and proper activation in order to help suppress the immune system. So we've been able to observe that HRV1A and 16 bind to their respective receptors, results in differential activation of the p38 MAP kinase pathway, as well as differential phosphorylation kinetics of the P65 transcription factor, and finally, that this resulted in differential gene expression not only between the two viruses, but between the different serums. In summary, we can basically say that yes, macrophages are being activated once HRV1A and 16 bind to their respective receptors. that there's differential phosphorylation kinetics occurring between the transcription factors and the intracellular kinases. Also, that macrophages will elicit differential cytokine production, that RT-qPCR data confirmed our RNA sequencing data, and finally, that in the case of rhinoviruses, that there is just not a general response. So in conclusion, macrophages exposed to rhinovirus do appear to contribute to the inflammatory microenvironment, and secondly, that the initial signaling events associated with rhinovirus binding to their receptors are different depending on the receptor used. I would like to acknowledge all these individuals, my predecessors, including Chelsea, who's here at the back today. Dr. Dave Hall, thank you so much for allowing me to have this opportunity to conduct research with you throughout my years at Lawrence. Dr. Sean Smith, who has shown me the interdisciplinary nature of my research through asthma epidemiology. Bryce Schiller, who also assisted me in better understanding my research. Sally Bourbon, who passed away last year. We do not want to forget her. She has contributed immensely to our research. And also Daniel Martin, who's currently working in Stockholm. The Dr. Rakinyala lab, which includes Michael Schreiber, who's also here now in our lab conducting research, Verdict's lab, and finally, the pulmonary medicine and allergy research groups. Questions?
You want to ask her tough questions because she's going to a meeting next week. And she'll get tough questions. So let her have it.
There's two questions. First one, why are you using bovine? Like, why not mice, which people like to look at compare humans to mice?
Oh, okay. That's an interesting question. We're looking at human macrophages because we want to make a direct connection with how it is that it can assist in how we can look at specific studies and how it can help with asthma exacerbations. So it really helped. There is a method in which you can actually extract upper respiratory tract epithelial cells from humans, but we wanted to look at the most common inflammatory cell that is within our upper respiratory tract. So we looked at macrophages in humans, which I think is better than looking at mice. So I've actually donated my own blood, obtained blood from Professor Dave Hall and other people on campus who wanted to assist in doing our research.
Cool. So is there a can you not do that with mice, or is it not as? Are they different?
I'm not sure. I don't think I
Mice
really.
do not get the human common cold. They will not bind to the receptors she described.
That's good for mice.
Oh.
That's good for mice.
And also bovine serum is one of the most common serums used in labs, and so we wanted to not only look at bovine serum but in human serum as well, which is more reflective of our inflammatory environment. Yeah.
So one more question. On your slide where you started looking at the MCP1 and you compared it to the CCL18.
Okay.
I think there's some data. If the bar graph that. Yeah, those two. Okay. Now, the orange new one. Okay, so on this one, both of them.
Yeah.
I'm noticing that the error associated with this data is much higher, I think, than the one on the next slide.
Than this slide.
Yeah. I'm
Yes.
curious why.
Well, the whole point was that this ELISA, we only did it 5 times, and so it was not statistically significant, so we actually had to do it like 30 more times in order for us to observe a statistically significant difference. And I just wanted to show an example of some actual statistically significant differences because we have a very small p-value. Yeah.
Sorry.
No, it's okay.
So I have a question that's more related to kind of your first couple of slides.
Okay.
I think, and I'm not sure if I missed it, but so you were relating how lower income groups are more prone to asthma and how that is increasing,
Yes.
right?
Yes.
And I guess that in that, I didn't understand how that links to looking at the rhinovirus itself.
Okay, yes.
Because I look at that and because of my experience, I'm like, well, low-income areas have a lot more PM2.5, and I'm looking at air quality issues there.
Yeah.
So I'm just curious of how direct that link between low income and more flu, or over time has this flu increased? Because I can do that for PM2.5.
Right, exactly. And so, well, my research with Dr. Sean involved just specifically looking at trends. And so we were just hoping in order for it to be like an overview, and then we can possibly in the future look at specifically how it is connected with those individuals having to be hospitalized due to asthma exacerbations. And we're just basically wanting to show that that there are certain socioeconomic factors that are associated with asthma exacerbations, and specifically hospitalizations. So there's not really a direct connection. In the future, we could possibly look at how it is that, why is it that those individuals are being hospitalized more, for example, and if it has to do with possible geographical location, for example.
I mean, I think there's quite a bit of work that relates that very directly to air quality issues.
Oh, okay.
Yeah, there are some air quality issues out there that out in my backyard puts it into lower income areas. And those lower income areas then have the impact of the pollution.
Yeah.
And so I just was curious if there's anything like that relating it to the rhinovirus.
Specifically the rhinovirus? We did actually find some data saying that it's generally associated with a specific type of rhinovirus, but we did not really look into that. But that's something I could do in the future.
Do you have a comment on the income quartile for the blue one, the Asian?
You can observe that it's actually increasing, right?
Yeah.
Depending on, you're seeing that
So you'd be healthier if you were poor in that case?
well, our conclusion was,
Which tag word of Walgreens you go?
well, I guess, I mean, it's possible that these people, that specific race group can afford healthcare more because they belong to a higher income quartile, and so they're being hospitalized more. And then one thing that we also discussed was possibly that Hispanics and African Americans belonging to a lower income quartile, the reason why we're seeing them being more hospitalized is because possibly they can't afford certain medications, and so they wait until the condition worsens before they're able to get treatment.
And this is for U.S.?
Yes, this is the U.S.
Is there a demographic, geographic demographic?
Yep.
That might relate to this?
Demographic?
As well?
Yes, there is.
I was thinking of the Asian population in the high smog zone.
Pardon? Can you repeat that?
The Asians in Southern California.
What about the Asians?
Well, they're in Southern California. They're smart. They're Asian. And I would not instinctively put them at the bottom of the socioeconomic heap.
Yeah, so there are various limitations of our research. We can account that result in limitations of our result, I guess. So.
Can you break it down by hospital? I guess is the kind of question.
Yeah.
So
Okay.
asking from a geographical standpoint,
Yes.
can you overlay hospital admissions from various parts of the country with pollution and things like that, whether it's rural or...
Yes, you can also break it down as well into specific geographical locations. But I'm not exactly familiar with how it is. I'm not exactly familiar with specific trends.
It seems to be income rather than genetic.
Income rather than genetic.
Yeah.
Yes, and that's something that we can look for in the
that
future
case,
as
big
well.
blue.
Yeah.
You talked about how most labs focus on epithelial cells.
Yes.
So why did you guys decide not to do that?
Because we wanted to see exactly, because asthma exacerbations are a result of a production of inflammatory cytokines, and they're mainly produced by macrophages, which are not epithelial cells, but they're immune cells that line our upper respiratory tract. And so we believe that it would have more of a direct effect as opposed to epithelial cells.
That is horrible to get students scooped.
Yes.
Everybody else is doing it. And
All right.
thank you very much.